Hi, everyone. Welcome to our webinar from Young Survival Coalition. We'll just give everybody a few minutes to go ahead and join, and then we'll start in about 2 minutes. So go ahead and join. You can introduce yourself, say hello, say where you're from in the chat, and we're happy to meet everybody. All right, it looks like the bulk of our people have joined, so I just wanted to introduce myself. I'm Jennifer Elliott, and in addition to being a breast cancer research and policy advocate, I'm also a breast cancer survivor, and I'm a graduate student concentrating in health policy at Rutgers University's Blaustein School of Public Policy. Today, we're excited to welcome Benjamin Gordon in a special Meet the Researcher presentation on triple-negative metastatic breast cancer. But before we start, I have some instructions for our audience. We want you to please use the chat to say hello and where you're from. And you may also ask questions in the Q&A. If you look at the toolbar at the bottom of your Zoom screen, you'll see a little Q&A, and you can put your questions in there on the bottom right. And go ahead, if you see questions in there that you'd also like to sort of upvote, you can go ahead and do that so we can be sure and answer them. And this session is being recorded and will be available on YouTube tomorrow. Another sort of bonus about today is it's actual National Cancer Research Day. So yay for people like Ben who are researching metastatic triple-negative breast cancer for us. And let me introduce Benjamin Gordon a little bit more officially here, although I've been calling him Ben between now and then. So Ben is in his 5th year of the University of Illinois at Chicago's 8-year medical science training program, which means he will graduate with both an MD and a PhD. He studies triple-negative breast cancer metastasis in the lab of Dr. Jan Kitayuski. Did I pronounce his name correctly?
Yeah, that's pretty good.
Excellent. Specifically, he's focusing on how tumor cells exit the vasculature during the metastatic process. He's a recent recipient of an F30 fellowship grant from the National Cancer Institute. And outside of the lab, Ben takes an active role in medical and science education and patient advocacy. So Ben is going to start his presentation now, and we'll take some breaks for some questions. And then at the end of the presentation, we'll have a really nice big audience question and answer session. So take it away, Ben.
All right, well, thank you so much for that introduction, Jen. And thank you so much, Young Survival Coalition, for having me. I'm extremely grateful to be here and super excited for this. Just before I start sharing my slides, I thought I would take a second to tell everyone how I became involved with patient advocates and what brought me here today. So as Jen mentioned, I'm an MD PhD student. So the goal is to become a physician scientist, both treat patients and conduct research and hopefully bridge the gap between the two. But in the lab during my training, and I'll show the training pipeline in a bit, I started to realize there was this, even though my goal was to help patients, there was this big disconnect between what I was doing and seeing the faces of who I was trying to help. So namely the cancer patients that I was doing research for. And that all changed. We have a program at UIC called the UIC Patient Brigade, which is a group of current patients and patient advocates that are involved in the research program here. And we gave a tour of our lab to a group of the Patient Brigade. And among that group was a patient advocate, a research advocate. Some of you may know her, Kimberly Richardson. She's kind of a rock star on the national stage, and she just was recently awarded the Stovall Award for her research advocacy and patient advocacy. And I saw this as a great opportunity to work with patient advocates and kind of bring a greater meaning to the work I do in the lab. And this led to what's called the Survivors Advising Scientists Educational Program, which is it's a bi-directional communication between researchers, young researchers in particular, and patient advocates to teach scientific concepts in a way that is bidirectional. So I'll talk about this more in a minute. But there are a series of educational videos geared towards cancer patients and advocates. So to help everyone become well-versed in the science and the science terminology. So it kind of brings everyone to the table so we could discuss the science, discuss the research, and help everyone become part of the conversation. My connection with Kimberly is what brought me to Jen, and it's opened up a lot of opportunities. And I just want to say this off the bat that I am looking forward to continuing working with patient advocates and patient advocacy throughout my career. And since I'm early on in my training, I welcome the opportunity to learn more from all of you and work with all of you. So let me start my slides. As Jen mentioned, it'll be broken up into parts. We'll do some conversational bits in between each part, and then the question and answer at the end. So briefly, I just wanted to show a picture of Kimberly, what I just mentioned. There she is touring the lab. And then here's a little shout out to the program I mentioned, Survivors, the Bioscientist Educational Program. The cartoon on the left is kind of the paradigm, the model we went after. So the trainees would create educational videos. We would get direct feedback from cancer patients and advocates, and then we would edit based on what they saw was confusing or needed clarification, and then we edited it and then we published those. You could scan the QR code there with your phone if you want to see these modules, or you could email sesep@uic.edu if you want to learn more. We cover topics on cancer biology, scientific experiments. We're going to go into clinical trials, and then specific videos on ovarian and breast cancer. So we're going to break this down into thirds. The first is kind of bring a face to research. What is it like? What's involved in biomedical research? Then I'm going to talk a bit about the biology of triple-negative breast cancer and the biology of metastasis. And then lastly, we'll go into current and emerging trends in terms of treatments. So as I mentioned, I'm an MD PhD student. Let's turn the laser pointer on. And it's an 8-year program. So after your undergrad, you train both in science and medicine. I'm right here in this whole timeline. So I'm in the PhD phase. And then the PhD phase is followed by my clinical clerkships, which I'll get, another 2 years, in which I'll get my MD. So at the end of it, I'll have my MD and PhD. And then following that will be residency, fellowship, and then faculty positions. And these are the physician scientists that are conducting research, running clinical trials, and trying to bridge the gap between science and medicine. Another reason why I bring this up is just to show you where I'm in my training. Even though I've learned a lot up until this point, I have not really dived in deep to my clinical care yet. So I'm not here to give medical advice to everyone, but to kind of talk about the current trends in research and bring a connection to what I know about the clinical connection. So what does a lab look like? These are pictures from my lab. I work in the lab of Dr. Jan Kidieski, and labs come in all shape and sizes, but typically they have undergraduate students from college that are just getting their feet wet and research. There's technicians that provide support, and a lot of times they conduct research experiments on their own as well. Graduate students, I'm a current graduate student, so that would entail me. Postdocs, so people who finish their graduate work and then go on and conduct more research within a lab. And then there's senior scientists, and then the principal investigator that is essentially the boss and kind of runs the program. So what types of research are there? So everything in blue deals with clinical research, dealing with humans. And then there's basic science. Right now I'm conducting basic science. And I bring this up because basic science is usually the foundation of what happens on the clinic. Sometimes there's a time lag between basic science and clinical trials, but usually there has to be some kind of foundational theory or science that gives way to the clinical work. And the Gold standard for testing if a therapy works or not for human participants is a double-blinded controlled clinical trial. So you'll see this at the top of the pyramid. It kind of goes into increasing levels of quality of evidence. But clinical trials are not the only types of clinical research. There's also cohort studies. So you kind of stratify patients based on a risk factor or a treatment they got, and then follow them either forward or backwards, either prospectively or retroactively. And then there's case control trials where you have an outcome and then you kind of do the opposite. You look for a risk factor or a treatment after you already know the outcome. And I bring all these the connection between basic science and the human trials up to kind of give face to what physician scientists are trying to do. We're trying to bridge the gap between what we're studying in the lab and what's going on in this blue pyramid up here. And a lot of that is bridged between what's called translational research. So that's usually done in experimental animals, and that's usually required before you could bring something to a clinical trial. So a recent example are the COVID vaccines. The basic science would be understanding mRNA, and the translational research would be testing the vaccines on mice, and then they were brought into human clinical trials. So let me stop there for now. And
so Ben, before you stop sharing slides, I have a question about one of them. If you go back a slide.
Mm-hmm.
So I wrote down, so you've got translational science on the left there with that nice arrow, and the basics and translational science starting at basic science and going up to medical guidelines. But how long does that actually take? Can't even guess. How long does that take?
Yeah, so it usually, it could take many years, even decades. I would say the recent example with the COVID vaccines was kind of accelerated based on the national emergency or the global emergency, where the transition for the experiments from translational to clinical trials happened all within a year. But many times it'll take a decade or more for it to go from bench in the lab to the hospitals.
And sometimes you'll hear that process of going from bench, what you're talking about in the lab, to bedside, right? So bedside meaning to the patient. So that bench part where you're still working on it, and then the translational part is taking it to patients.
Correct.
So I have that.
Yeah. So yeah, that's a common mantra in for physician scientists and MD PhD programs where we say bench to bedside. So bringing what we learn on the research bench to the hospital bed.
And does it ever go backwards then? I mean, I hate to say backwards because you're never in science, hopefully going backwards, but does it ever go back to the bench after you've tried something in a patient? Can it go back to where you realize you figure something else out?
Yeah, so it's very often a cyclical process because a lot of times you have the basic science and the translational research and the clinical trials, but the clinical trials, eventually you have enough of them that are positive, will give rise to medical guidelines. But a lot of times there are more research questions that come from the clinical trials. For instance, why is the drug only working for a certain subset of the patients? Why didn't a drug work at all? And a lot of times, especially for physician scientists that are kind of bilingual in both the medicine and the research, will see the new questions that arise from the clinical practice and will create more hypotheses for the basic science. So they'll have a new question that are based on observations in the clinic, and that will help inspire more basic science experiments to kind of address those. And then an extension, more translational research.
And is there a point where patients or advocates can have a voice in this type of translational research?
Yeah, so one of UIC's cancer centers, big sayings is from instead of bench to bedside, they say from community to bench. So that basically is an extension of that, meaning that we want the community to have a voice in this whole process. And I think that's very important because there's a history of scientists and doctors making decisions that don't have the input of patients, and there's a lot of oversight when that happens, where we're not directly addressing the needs and wants of the community that we are supposed to serve. So there's many different areas where patient advocates get involved. When I submitted my fellowship, I had Kimberly, who's a patient advocate, read it over, give her input, and write a letter of support. So my application process did include a patient advocate, but just showing up to conferences to give your input on the research, show either it's research conferences or medical conferences, grant review, paper reviews. I think there's many different ways to get involved in this process. Even if you're not at the bench doing the experiments yourselves, grant writing kind of influences the future of research, where it's going, and I feel like everyone could put their fingerprint on that.
And talk to me a little bit about that letter of intent. Letter of support, sorry.
Yeah, so for grants, you know, I was the principal investigator for my fellowship, but it's really a team you assemble. So you have a team of, you know, my sponsor, who is my boss, my PI. Then you also have, I had some clinical mentorship on the grant, and then I also had some other scientists that are advising me. But I also had Kimberly and patient advocacy as part of my training plan for the fellowship. So patient advocates were a part of the team essentially for my grant submission.
So if a patient advocate contributes a letter of support, does that sometimes give you sort of a leg up in your research? So would that then inspire researchers to search out patient advocates, hopefully?
Yeah, I mean, anything that you write in a letter of support kind of is part of the proposal. So you're proposing to the government what you, or the agency, what you plan on doing. So if you say, I plan on including patient advocates in my proposal, then you're kind of committing yourself to including them in part of the project. So I think that's a good thing. It'll be ultimately up to the PI to hold themselves accountable to their stated goals. But I think if you state the goals and follow up on it, it kind of was a good thing.
That's great, thank you. Shall we move on to your next part? Okay. Unless there are other questions from other people, I think we'll just move on.
All right, so let's move into the biology of triple negative breast cancer, specifically metastatic triple negative breast cancer. And this part will be important for the last section as well, in terms of tying the basic science of TNBC to emerging trends and treatments. So let's take a step back and define triple negative breast cancer. And you know, many of you will already know this, that it lacks the hormonal receptors and HER2 amplification. And these count for about 15 to 20% of breast cancer cases. And this is troublesome because without these hormonal receptors or HER2 amplification, there are less targeted therapies in market compared to the hormonal receptor positive cancers. However, this definition based on these statings of these markers are just the tip of the iceberg, because what we're learning in both the clinic and in the lab is that TNBC is much more than these three stains. TNBC is a very heterogeneous disease, and that's just a fancy way of saying that everyone's cancer is different. And there's many different ways we could stratify TNBC, and each person's disease will essentially be its own disease. And I'll kind of go into that a little bit more here in terms of classifying TNBC. So first of all, let me turn my laser back on. If you look in the middle, this is a terminal duct lobular unit, and that is the duct where milk is produced in the breast. And there are two cell populations that I want people to focus on. We have the luminal cells in the middle that produce the milk, and then we have the myoepithelial cells, or there's some kind called basal cells that line the milk producing cells that help squeeze it out. And I bring that up because sometimes breast cancer is talked about in terms of its intrinsic subtypes. And I think an easy way of understanding this is how much do the cancer cells resemble the native cells, the normal cells of the breast tissue. So the basal type intrinsic subtype will resemble the myeloepithelial basal cells found in the normal breast tissue, and the luminal type intrinsic subtypes will resemble the milk-producing cells, and these are done by certain cell markers. About 70 to 80% of the basal type overlaps with triple negative breast cancer. So sometimes doctors and scientists will interchangeably say basal breast cancer or TNBC, triple negative breast cancer, but it's not always the case. It's just most likely the case that TNBC and the basal type will overlap. Another way of stratifying TNBC is to look at hundreds and hundreds of genes through sequencing and looking at the genes that are turned on and off and kind of weighting that all together and coming up with stratifying it into molecular subtypes. And this was done most prominently about like 10 years ago, starting like 10 years ago. And you can see some of the subtypes here. You have basal-like 1, basal-like 2, immune modulatory, and mesenchymal-like, luminal androgen receptor. So, you know, you can think of genes as traits. And if you look at hundreds and hundreds of traits, then you start getting a personality. So you can think of these subtypes as the overall personality of the cancer. It kind of gives a wide-ranging look at the different characteristics. So it's another way of kind of subtyping TNBC. And I'll bring up these subtypes later on when I talk about chemo resistance and other types of therapies, because this type of stratification will hopefully one day help us decide which kind of therapies work for a certain subset of patients. And then another way of classifying TNBC is through specific genes, proteins, or mutations. BRCA1 and BRCA2 are an example of this, and PDL1 expression is another example. And again, I'll come back to these concepts. And I just want to mention that this is not an exhaustive list. There's other types of ways of classifying TNBC, but this kind of gives everyone a taste of what's been going on over the last 10 years in terms of trying to further dissect TNBC. Moving on to metastasis, because this is a talk about metastatic TNBC. I want to say that metastasis is a highly complex cascade with multiple steps. It's actually a very inefficient process. So a lot of cells try to complete the metastatic cascade, but they often fail. So it's only very few cells that actually do end up completing this. And we'll start here on the left. This is the primary tumor site. So this will be in the breast for this talk. Usually it starts off with invasive-like characteristics of the primary tumor. Usually a process called angiogenesis or vessel growth also occurs at this stage, which is needed for the cancer cells to invade and eventually enter systemic circulation. So that process is called intravasation. So here you see the cancer cell entering the bloodstream, and then the cancer cells have to survive in circulation. A lot of them will die at this step, but the few that are successful in metastasis will survive in circulation. And then there's a step called extravasation, which is the opposite of intravasation. This is the exit of cancer cells out of the bloodstream into the site of metastasis. And then the cancer cells here in red, will form micrometastasis and then eventually colonization if there is a metastatic colony. I earlier said that I perform metastatic TNBC research in the lab of Dr. Jan Kudieski, and I wanted to briefly give you an overview of what I study. I study one of the steps called extravasation that I just mentioned. So we're interested broadly on how tumor cells interact with the vasculature, specifically for the step. So tumor cells exiting the vasculature before making metastatic colonies. So one of the ways I study this is try to model what's happening in this process in the laboratory. So this is outside any organism. This is in a microchip. And what you'll see here on the right, I'm about to show you a video, is TMBC cells, metastatic TMB cells in red, in a model that's trying to capture a blood vessel and having cancer cells exit. So you'll see the cancer cells in red flowing over a vascular wall, which you'll see in gray cobblestone in the back. And we'll start this video. So here we're flowing out the red labeled breast cancers, TMBC cells, as if they were in a blood vessel. And then you'll see here that some of the cells started sticking onto the vascular wall and will eventually move from the top layer underneath. And my lab will perform genetic studies, will knock out certain genes or will target certain genes to try to figure out what drives this process. So that's just essentially the nuts and bolts of my research. You could always contact me if you want to learn more about exactly the genetic pathways I study. But I just wanted to give an example of one way metastasis could be modeled in the laboratory. Another way, method that I use outside of the in vitro stuff, so in vitro means basically in a petri dish, we also use mice as model organisms to look at the extravasation step. So here is the inside of a mouse lung, and the green is the vasculature inside the lung. So they're capillaries of the mouse lung. And in red, again, is the cancer cell. And you could see here is a cancer cell that has exited the vasculature. So it has completed extravasation and it's now kind of wrapping its way around the capillary. So we'll take another break here and see if there are any questions before we move on.
So we have one question that just popped up in our Q&A, and I just want to remind us to use the Q&A section, which is the far right of your toolbar, to pop them up. Sadly, I'm not good enough to follow questions in chat and Q&A. So if we just keep them to Q&A, that'll help me out a lot. So we have one question, and this is a great question. I really appreciate questions coming in, and I know Ben does too, because it helps us just too. It helps him in lots of ways to sort of refocus and make sure that he's studying things that people are interested in and that you feel are helpful. So here's our question. It says, what's the hope with your study, Ben? Do you hope to be able to stop colonization of cancer cells? Do you hope to stop metastasis? What's the sort of end goal hope?
Yeah, so we study a genetic signaling pathway called the Notch signaling pathway. And there's a specific gene within the pathway called JAGID1 that is correlates with poor prognosis and increased metastasis. So I'm studying a gene that's involved in increased metastasis in TNBC patients. And one of the ultimate goals is to be able to target this gene if the patient presents with upregulation of the gene called JAGID1. And the step I'm interested in studying that I just showed is extravasation. So the ultimate goal would be to target JAGID1 to prevent the exit of tumor cells from the vasculature and hope they just die within the cancer cells just die within the vasculature instead of exiting.
So I'll ask a follow-up question to that. Do we know how exactly they pop through the vasculature yet? I say we loosely. Do you know?
That's a very good question, and it's what I'm researching now. So a lot of what we theoretically based our knowledge on of tumor cells exiting the vasculature is based on white blood cells, so the cells of the immune system. So you have white blood cells circulating in your blood circulatory system all the time, and then you have a cut or an infection, they have to exit your bloodstream and have to enter the infected tissue or the cut or wherever the cells need to go. The study of white blood cells and inflammation exiting the vasculature is very well known, very well regulated, and we know what proteins and genes are involved in that cascade. There's been a lot of parallels between cancer and white blood cells in terms of this process, and I'm trying to see to what extent cancer cells parallel white blood cells. It's from my studies, it seems like there's some tools that the cancer cells do borrow from the white blood cells, but there also seems to be unique properties of cancer cells exiting the vasculature. And those unique properties might be able to be targeted one day to uniquely target cancer cells without affecting the migration of your white blood cells out of the vasculature.
So that sounds like almost a catch-22. We need inflammation in our body. Inflammation plays a good role in the sense of healing, helping to heal our body, but also could play the other side of the sword, which is helping with metastasis.
Yeah, and you know, it's a common theme in cancer biology is like, how do you target the cancer cells without affecting beneficial aspects of your normal homeostasis or your normal body functioning. So that's where finding maybe unique roles that TNBC uses, such as the GNI study, JAGED1, in terms of increasing extravasation, the exit of the cells out of the bloodstream might be useful.
Okay, thank you for that. We have another question from our group. And this goes back to when you're talking about the subtypes of triple negative breast cancer. And this is again another really good question. And again, we really appreciate your question. So why aren't subtypes? Yeah, I'll let you get back there before I ask. Yes. There we go. So why aren't subtypes used and discussed more in the clinical setting?
Yeah, so I think it's a very good question. And I'm gonna address this more in the third section, but I'll briefly touch upon this now. So at UIC at least, and again, I'm not a clinician yet, so this is based on talking to some fellows at our cancer center, is that typically we will look at PD-L1 and BRCA here in terms of classifying TNBC. Obviously, the research is ahead of just BRCA and PD-L1. There's many other subtypes, and the short answer is that there are a lot of studies and clinical trials right now looking into the subtypes and trying to match the treatment with the subtype. But whoever asked the question is correct that it's not commonplace. It's not the standard of care right now in terms of sequencing everyone's tumor. And you know, that might change over time based on the results of the clinical trials at big academic centers.
Thank you for clearing that up. Shall we go on to your next section?
Okay, so now we'll try to tie everything together. And I think the question about the subtypes is great because it's going to be an emerging, a constant theme throughout the current and emerging treatments. So first, let's start with chemotherapy. And for metastatic TNBC, that includes the taxanes, paclitaxel, anthracyclines, platinum agents, and cyclophosphamide. And when I go through the different current and emerging treatments, I'll try to kind of guide everyone through where the active research is and how it ties into the research. So first of all, to bring in back to the subtypes, let me turn my laser back on. I have highlighted in pink underneath each chemotherapy the subtype of TNBC that has been shown to best respond to the specific chemotherapy. So for taxanes, it's the basal-like intrinsic type. Anthracyclines, there's been shown to be a higher response rate for basal-like 1 and mesenchymal-like. And then platinum agents, there's BRCA mutation status, have better response, and so does a basal-like 1 molecular subtype. And so the big area of active research is to kind of base the chemo response based on the molecular subtype. So kind of matching patients to the right treatment based on their subtype. What's highlighted in pink, on 1 second. What's highlighted in pink was done from cohort studies. So those were done retroactively, looking at stratifying patients based on their risk factors. So their subtype, and then looking retroactively how they responded to treatments. Another big area of active research is chemotherapy resistance, and there's many different areas of that, such as drug efflux and stem cell-like cancer cells that researchers are looking at right now in the lab, and trying to understand why certain patients have resistance to chemotherapy. Because right now, I should have mentioned this from the start, only about like 40% or so of patients do respond well to chemotherapy treatments. And a big area of active research is to try to understand why patients are not responding and which patients would best be matched up with certain different types of chemo options. So I'll talk about this in a bit, but this is where subtypes might be helpful in terms of coming up with treatments. Then the targeted therapies, a lot of these are emerging paradigms, emerging areas of research and emerging treatments, because the chemotherapies have been around for a lot longer. And I've broken this down into angiogenesis inhibitors, PARP inhibitors, and immunotherapies. And I will kind of talk about these more in detail in the upcoming slides. But again, for this is the active areas of research are predicting and increasing the responses and based on up and coming research. And other areas of active research is finding additional targets. So what besides these could we possibly target? As I said, I'm looking at the protein called Jagged1. Will that be a future targeted therapy? I mean, the jury's still out on that, but there's other areas of active research that are continuing to look for more targeted therapies. So let's go over each one by one. So angiogenesis, I brought up earlier, is the process of growing new vessels from previous vessels. And both the primary tumor and the metastatic cells need to do this in order to grow and survive. You may have heard of VEGF. That's a very that's the most common targeted angiogenesis factor. It promotes angiogenesis, and you can see bevacizumab is the common treatment for that targets VEGF. And these are currently under clinical trials, and the problem is that a lot of patients eventually will build resistance to angiogenesis inhibitors, because one of the promoting factors of angiogenesis is hypoxia, the lack of oxygen. So sometimes when you target angiogenesis and increase hypoxia, that actually increases the stress to create more vessels. So kind of understand this process will maybe help come up with more successful angiogenesis strategies. Another type of targeted therapies are the PARP inhibitors. And I'll kind of walk you through what these do. So PARPs are proteins involved in repairing single-stranded breaks in the DNA. So DNA is a double helix structure, and when one of the strands breaks, you need a PARP to help repair it. BRCA1 and BRCA2 are proteins involved in double-stranded repairs through a process called homologous recombination. So what happens when you have a normal BRCA status is that if you inhibit PARPs and you have these single-stranded breaks, and then eventually you have enough breaks that you have a double-stranded break, if your BRCA is intact and working well, it will repair the DNA and the cell will survive. People with germline BRCA mutations, either 1 and 2, the same thing will happen with PARP inhibitors, that you'll have a single-stranded break, and over time, since you're inhibiting PARP, you'll have double-stranded breaks. And without BRCA1 or BRCA2 there to help fix the double-stranded breaks, the cells will eventually die. And that's the kind of theory behind drugs such as olaparib and olaparib, which are PARP inhibitors. And then to kind of bring this back to the emerging subtypes and research, in addition to germline BRCA mutations, you also have what's called BRCAness, which is mutations of BRCA1 or BRCA2 that you develop later in your life, or types of characteristics that kind of resemble the inability to repair double-stranded DNA breaks. So other proteins involved in double-stranded break fixing or mutations you develop to BRCA later in your life. So here's a recent article, and we're trying to understand the difference between germline BRCA mutations and BRCA-ness. It turns out there seems to be variability in the response to PARP inhibitors, whether you have germline mutations or have BRCA-ness in the tumor. So trying to understand why BRCA-ness responds differently to drugs compared to the germline mutations is an active area of research that we're still trying to understand. Next, we'll move on to immunotherapies. The big ones that I'll talk about are pembrolizumab and atezolizumab. And these are all over the news right now. So I'll first kind of briefly mention how immunotherapies work. So so here shows a dendritic cell and a T cell, but this is also applicable to tumor cells for the purposes of this talk. So a lot of tumors will express PD-L1 and PD-1, and it's basically and you probably also heard of CTLA-4. They're essentially breaks of the tumor cells saying, hey, don't destroy me, I'm one of you. And you have cells throughout the body that have this message of PD-1 and CTLA-4, so you don't have autoimmune disease. You basically, your cells of your body tell your immune system, hey, I'm one of you, don't attack me. But the problem with when you have cancer is that the cancer cells could do that too, is, hey, immune system, don't attack me. What pembrolizumab and atezolizumab do is block the signal. So they block either PD-L1 or PD-1, and they help the T cells destroy the tumor cells. And then to bring up some current events, so recently Roche updated its indication for Tecentriq, which is an inhibitor of PD-L1 for metastatic triple-negative breast cancer. So for the Impassion trials, they had one trial where they showed successful results in progression-free survival, where they mixed PD-L1 and nab-paclitaxel, which is a taxane. But then they followed up with another trial, another phase 3 trial, where they, instead of using nab-paclitaxel and atezolizumab, they used paclitaxel and Tecentriq, and that showed no benefit to the patients receiving Tecentriq. So, and when you look at both those trials, you can say there's mixed results because one worked and one didn't. But Roche voluntarily removed their indication status based on what was happening globally with the market, because if you look at the left, Keytruda has been recently approved by the FDA as a combination therapy with chemotherapy for metastatic triple-negative breast cancer. So I thought I'd bring this up to kind of get the latest updates on the PD-1 and PD-L1 inhibitors. So this is my last slide before we could do more of a question and answers session. But I just wanted to bring up two emerging trends to kind of tie everything together. So one's the Future trial, which recently took place. And this kind of answers the question that we got at the last break, is why doesn't everyone get their tumor sequenced and to help come up with the best strategy? This clinical trial tried to do this with 69 patients. So this was a phase 2 trial. So only a limited amount of patients, but they looked at 400 genes, I believe, and also did the staining of the TNBC, and they broke it down to molecular subtypes. So I brought this up before. There's the luminal androgen, the immunomodulatory, the basal-likes, the mesenchymal, and they further stratify this in BRCA mutations or AKT pathway mutations. And then based on the status of the molecular profile of these cancer cells, they would match the patient with the therapy. So basal-like, which I mentioned before, if they had the BRCA mutation, would be matched with the PARP inhibitors. That makes sense because the BRCA germline mutations are already matched with PARP inhibitors. If they didn't, they would give anti-VEGFR, which is the angiogenesis inhibitor. Immunomodulatory means that the tumor cells has a lot of immune cells infiltrating, a lot of immune signatures. So that's why they use the anti-PD-1 and paclitaxel here. So this is the kind of emerging trends in precision medicine. What people call this precision medicine is matching the patient's personal characteristics with the correct targeted treatment. And another important area in research that I think is very exciting is the gains we've made in understanding the tumor microenvironment. And I think this is especially important when trying to figure out why patients either respond or don't respond to immunotherapies. So the tumor is rarely just the cancer cells themselves. The tumor is a collection of the stroma, the vascular cells, and the immune cells, and the extracellular matrix, so fibrin and all these different proteins that surround the tumor. And the amount of T cells, for instance, so the cells that kill, the immune cells that kill cancer cells infiltrating the tumor microenvironment plays a really significant role in whether they're going to respond or not to immunotherapies. So a lot of great strides have been taken to understand the tumor microenvironment and how it might affect the treatments. And it also might explain why certain therapies might work in a petri dish, but don't work inside the tumor microenvironment, because the petri dish doesn't kind of encapsulate all the things that are happening within a tumor and all the different other cells that are interacting when a drug is introduced. So again, I just want to thank you so much for your time. I'm looking forward to the question and answer session. Thanks, Young Survival Coalition, for having me. And I look forward to hearing and reading your feedback so I could kind of take that and move forward with it. So thank you.
Thank you, Ben, for your presentation. And we do have some questions in our Q&A section here. So we'll put you on the spot a little bit and see see if you can answer our questions. So one of the questions that we have is, how is BRCA-ness tested? And so we know there are two different types of BRCA-ness, right? There's the one that's sort of in your whole body that you're born with, which you alluded to before, but also the you can have your tumor can have BRCA specific qualities.
Yeah, so that's a great question. So initially, I think the idea of BRCA-ness came up. So as you mentioned, there's the germline mutations, and then there's different epigenetic or other types of mutations called somatic mutations that occur not when you're born, but later in life, that kind of gave rise to the idea of BRCA-ness. But BRCA1 and BRCA2 are not the only genes involved when we talk about BRCA-ness. There's other genes that do similar processes as BRCA1 and BRCA2 that could give rise to BRCA-ness. And these are genes involved in double-stranded breaks, just like BRCA1 and BRCA2. And then there's other genes that are involved in DNA breaks called such as ATM and RAD51C that researchers look at. But the emerging trends for research is to do next generation sequencing and looking at a whole wide range, sometimes hundreds of different genes, and trying to get a look at the landscape of the DNA damage and repair response.
So is there a test at this point or for BRCA-ness that you know of?
So I'm well versed in what, so I highlighted an article that did next generation sequencing for patients, but that's definitely not the standard of care right now. I don't know the clinical standards for BRCA-ness as of now. It might be different from institution.
So it's possible a person who could find out if there's a clinical trial at their treating institution by asking their oncologist, for instance, if there's one.
Mm-hmm.
Would that be a way?
Yeah, for sure. I definitely, for, that's beyond just the BRCA-ness too, for any of the molecular subtypes or anything I mentioned, I definitely recommend talking to your oncologist in terms of getting paired with possible clinical trial or just seeing what your options are. Your doctor will obviously know most about your cancer characteristics. So that's definitely the person to first reach out to.
Great, thank you for that. And so sort of along the lines of testing your tumors, we have a question about, is there a way to predict which patients will be chemo resistant? Is there a current test, or is that still in trials as well?
Yeah, so this is all emerging research. So maybe if I could go back to the screen, I could go back to my chemo slide.
Absolutely.
Yeah.
We love images. Those are helpful.
So what's in pink under each chemo is some molecular subtypes of TNBC that best predicted response to the specific chemotherapies. However, these weren't experiments that were designed specifically for the subtypes. They were done retrospectively in a cohort study. So what they did is they looked at 5 different clinical trials that were already performed. They looked at the patients that were basal-like 1 or mesenchymal-like, or CBL1, and then they looked backwards and saw like which patients responded the best, and then they made some conclusions from that. So basal-like 1 and basal-like 1 responded really well to the platinum agents and anthracyclines compared to the other molecular subtypes. And then when I talked about the future trial, that's another way of doing it. Is those are clinical trials where they first look at the subtype of the patient and then match them with the therapy in real time, because that's as opposed to this, which was a retrospective analysis. So a lot of the research looks retrospectively, and then the emerging areas of research are trying to apply this to patients undergoing current treatment. So it's still a work in progress. It hasn't totally met fruition yet, and the jury's still out if these types of targeted precision medicine will actually work. But it's in the pipeline.
So is this going back again to asking your oncologist if there are trials that you could participate in again? I feel like that's our theme for this part.
Yeah, so it's definitely worth looking into what clinical trials are available and checking to see, what the research design is for that clinical trial. Are they going to look at molecular subtypes or not?
Okay, great. Thank you for that. We have some other really good questions. Lots of good questions coming up. And since we only have 8 minutes left, I want to make sure we get to as many of them as we possibly can.
Okay, sounds good.
So I'm going to combine 2 of them because you just talked about some of the emerging research. But one of the questions is sort of, in your opinion, what looks most hopeful about that emerging research? And then the other sort of piece of that is, do vaccine trials, does that count as emerging research? Or is there sort of more, or are we further along with that piece of it?
So is that in reference to the current vaccine trials, or?
There is a current vaccine trial that was just FDA approved, correct? For TNBC?
Oh, so we're talking, okay. There's many, some reason
maybe
I
not just
COVID, yep.
That's fine. Maybe not just, and I'm speaking, perhaps someone can send me a message about whether it's been where we are in the approval process. I don't know if it's been approved that the trial is about to start, or I don't want to speak incorrectly and send any hope in the, hope for something that's not actually happening.
Mm-hmm. Yeah, so I wrote a review on the tumor microenvironment and breast cancer vaccines. It wasn't geared specifically to TNBC, but there are a bunch of clinical trials of TNBC breast cancer vaccines. So if you Google my name, Benjamin Gordon, and type in tumor microenvironment, that paper will come up. And there's in the paper, I list a bunch of current clinical trials ongoing for TNBC vaccines. In terms of emerging trends that I'm personally excited about is the ease in which we could kind of classify someone's tumor characteristics based on the molecular subtypes, as I just talked about, next generation sequencing is becoming cheaper by the day. So this ability to connect the science, because these molecular subtypes came out over a decade ago. And so the research was way ahead of what was happening on the clinic. And it's really exciting to see that we're able to take this theoretical knowledge and try to come up with treatments that are best suited for each patient based on their individual case. Another emerging topic, and I highlighted this in my last slide, is the tumor microenvironment. I think ultimately understanding the molecular profile of the tumor, but also understanding what's going on within the microenvironment, and be able to both target the tumor itself and what's going on in the microenvironment will be a two-hit process to kind of comprehensively attack the tumor.
Great, thank you for that. And I did get some clarification from Ali, one of our audience members, that the TNBC vaccine is phase 1 approved, so the trial's recruiting, I believe.
Okay, yeah, so then it's, I'll consider that emerging to answer the question.
Yes, very much. So again, we have lots of questions coming in, so I'm so excited. And so going back to tumor subtypes, and for TNBC subtypes rather than tumor subtypes, but specifically the subtypes of triple negative, do we know which are more likely to metastasize at this point, or are we still learning that?
Yeah, so there's different prognoses associated with each one. I don't have the statistics on me right now, but they were originally developed based on their genetic signatures. So the basal likes, for instance, are enriched in genes that control cellular proliferation, for instance, and that's why they might respond to chemotherapies that target the cell cycle mostly. And there's information out there about metastatic probability and prognosis, but I don't have those numbers in front of me right now. Okay,
then so this is another, thank you for being clear on that this is an example where we need to perhaps ask our oncologists again about specifically if you know that you carry a certain type of triple negative breast cancer, then you can specifically direct questions about likelihood of metastasis to your oncologist, or unlikeliness, because we like to think about that too. So talk to me about if there's any research focusing on the prevention of recurrence of triple negative, because it's more likely to recur than other types. I feel like that's kind of what you're talking about anyway, or what you're researching anyway, when particularly when you're talking about researching the vasculature and how it pops out and all of those sorts of things. But perhaps there's pieces of it that are more like sort of going in that direction as well.
Yeah, for sure. I appreciate you bringing up my own research, because it is true. So right now I'm in the early on stages of my project, so kind of studying the mechanism. So I don't really have a specific drug I'm testing to really try to prophylactically prevent relapse, but that is an ultimate goal, is either to prevent an early stage primary tumor from metastasizing, or preventing a case of relapse. Another example that comes to mind right away is, and someone else brought this up early in the question and answer session, is the breast cancer vaccines. That could definitely be thought of as a prophylactic measure in some instances. Cancer vaccines in general could be therapeutic, or they could be prophylactic or secondary prophylactic in terms of preventing relapse. So those clinical trials are underway with the breast cancer vaccine. So, and I'm sure there's other types of prophylaxis that are in study now too.
So there's so many good things happening now in research, and we're so glad that you are here to tell us about all of the things that you're working on. And we do, just to bring it back home to patients and advocates before we wrap up, I know you work with Kim, and there's that way that that patients and advocates are able to connect with researchers or through already established programs. But is there anything else you can think of, any ways that patients and advocates can connect with researchers, sort of move research forward?
Yeah, so maybe I'll bring up my slides one last time. And we'll go way back to the beginning. So a lot of the topics I brought up are very complex scientific topics, and that's one of the reasons why we created this educational programs to teach both how science is done, but also teach the biology of cancer. And so if you were to scan this code here on the right, you could see what we did already. And the goal is to help everyone become proficient in the language of science. So we could talk about the experiments, we talk about the results, and cancer patient advocates could become involved in the decision making that is usually reserved for select people. But I'm also listing the email down there at sasep@uic.edu. That's our email address. So if you have any follow-up questions or would like to become involved anymore, that's one mechanism. But there's also, that's not the only way. You could get involved at your local cancer center or your local academic institution to see what kind of advocacy groups are there. There's also advocacy groups on the national stage, and see what opportunities are involved locally in terms of grant review, scientific poster review, paper review. I think there's currently a lack of having enough inclusion, and I think there's so many opportunities for there to be a bi-directional communication between physicians, scientists, and the community.
Great, thanks for that. And I just want to give a, just in case advocates are unconvinced that there actually are patients that are involved in research labs, I'm actually involved in a research lab at the School of Public Health. So I serve, I go to all their meetings. I am able to give feedback at all levels of research, and ours is not clinical in the sense that we are not developing drugs, but it's more about survivorship care and end of life care. So there's all sorts of avenues to be involved in. And this has been so lovely. Thank you, Ben, for giving us lots of good info. Thank you to Young Survival Coalition for hosting our live stream for us. And if you are interested in other Young Survival Coalition programs or in support, you can visit youngsurvival.org. Also, a reminder that this is being recorded and it will be up on Young Survival Coalition's website starting tomorrow. And so you can view it at your pleasure. And again, thank you so very much for this. And we look forward to sort of learn, since you're such a young researcher, and I say young in terms of your professional career, and then we are looking forward to sort of what lovely things you bring to the triple negative metastatic community.
Well, thank you so much for having me. This has been a pleasure, and I look forward to continuing the dialogue and working with all of you.
All right.